Welcome everyone to the OHSU DMICE Research Conference. And we have tried to have some outside speakers in the pandemic. And we have such a speaker today, Dr. Tudor Oprea. Tudor was suggested to us by Guan Ming because they collaborate, but actually I know him well. He invited me a number of years ago to visit Albuquerque and the University of New Mexico, which is a lovely place to visit. Anyways, he is professor of medicine, pharmaceutical sciences, and chemistry and chemical biology, and also division chief for translational informatics at the University of New Mexico School of Medicine in Albuquerque. He has both an MD and PhD degrees from the University of Medicine and Pharmacy in Timisoara, Romania. He is a very accomplished researcher, has co-authored over 250 publications, has 10 patents, and has edited 2 books in his main area of focus on drug discovery. And currently he's looking at development of validated artificial intelligence models for target selection in drug discovery by combining numerical and text mined information to model health via knowledge graphs. And so his main activity is serving as PI of the Illuminating the Druggable Genome Knowledge Management Center, which is one of the NIH Common Fund initiatives. So he will speak to us. We'll hopefully have some time for questions. And I always enjoy these talks that kind of cover both bioinformatics and clinical informatics. And so we'll see some of that. So take it away, Tudor.
Thank you, Bill, for inviting me, and thank you, Guang Min, for inviting me. I have to say my first encounter with Dr. Hirsch was as a student in the AMIA 10 by 10 course back in 2012. I need to learn more about medical informatics, but without further ado, because I have a lot of material to cover, essentially what we try to do is insert informatics, data science, and machine learning in all the 3 pillars that I consider 3 pillars of drug discovery, which is diseases, targets, and drugs. And through the IDG program, we try to cover all these. So hopefully by the end of today's talk, I will be able to convince you that that is the case. So very briefly, the IDG started with an interesting comment from Al Edwards, who leads the Structural Genomics Consortium. He essentially pointed out that, wait a minute, 75% of research is done on the 10% of the genes that were known before the human genome was mapped. And this was 10 years after the project was completed. And now we're celebrating 20 years, and you see a lot of publications touting the successes of that, the recent analysis in Nature. In fact, all the Nature publications are running this type of analysis. The reality is we still have a lot more to cover. So hopefully I'll be able to convince you of that. My adventure joining the IDG started with a 7-year curation effort that ended up in this publication, which is currently my most cited paper. It's we essentially came up with a mapping of all the drugs to all targets, and in the end we covered about 1,600 FDA approved drugs and about 900 targets, of which 600 plus are human. And of course all that information is available in PubMed Central as well. So we then joined, based on this expertise, we joined the IDG consortium. The part that's highlighted in dark blue is the experimental section of the IDG consortium. We have a stellar group of scientists that work on G protein coupled receptors, ion channels, and kinases, and they generate experimental data, which gets processed via help from Stefan Schür and his team in Miami, and then gets deposited into our resources, which are then made available through the Knowledge Management Center. And we also have OHSU part of this proposal through a cutting-edge informatics technology tool. Obviously, in his case, it's the Reactome group. So what do we do at IDG is essentially to collect data as much as possible from across a wide spectrum of information sources. Large-scale experiments, those are given preference. And then we also map things like patents and drug labels. We're doing forays in the clinical trial area as well. And all that information gets standardized using a series of ontologies, including Monarch, which is spearheaded by Melissa Handel and her group. And all that goes into Target Central. And you can access Target Central through the PHAROS database, which is pharos.nih.gov. And more information about our consortium is at Druggable Genome.net. These are some of the resources we catalog. Of course, there's a lot of them, so I would not be able to do justice to all the information. Just very briefly, the ones that are marked IDG are things that we directly fund or support. And those that are in blue are what's considered to be other common fund projects. And all told, we have billions of data points, but when you look at attributes, we're currently at about 700,000 on a per protein association basis. And we have proposed a knowledge-based classification of proteins. So as probably most of you are aware, in bioinformatics, a lot of people spend a lot of time looking at the primary sequence and identifying motifs and then 3D structural classifications of proteins. What we try to address the question is how much do we know and what we know on a per protein basis. So we came up with 4 categories, T-clean. These are the mode of action targets. That was the subject of the Nature Drug Discovery paper that I mentioned in the first place. TKM, these are the proteins that are annotated with small molecules. T-bio is where we know most of the biology. And of course, the dark proteins are the ones that are understudied or just very poorly described. And so the inner ring shows the distribution of the 4 categories. And of course, wherever the function is known, we have that information as well, which is the outer. So how do we come up with the TKM attributes? We took all the approved drugs for which we have bioactivity data.
If you have a question on your fill, please press 1.
Okay, maybe people should be on mute. Was that question for me or should I continue?
No, I would continue. I think that sounded like the OHSU voicemail system. So okay. I don't know, someone must have had their phone unmuted. You can go ahead.
All right. So when you look at kinases, for instance, the cutoff is to cover the 90 percentile of the approved kinase inhibitors still is at about 30 nanomolars, whereas if you look at ion channels, the cutoff would be at about 20 micromolar. So therefore, we have this sliding scale on a per target class basis. The thing that we look at is the available publications. So with help from Lars Jensen in Denmark, we implemented something called a fractional publication score or paper count. If you say, for example, have 100 publications and you're interested in the insulin receptor, if all those papers only mention the insulin receptor and nothing else, then that fractional publication count gets to be 100. If you're actually looking at 100 papers that talk about 20,000 proteins each, then that is a very, very small number. So we adjust for that. We also take into account the gene reference into function set, which is maintained by NCBI. We also mine antibodypedia.com. Unfortunately, we don't have data on unique antibodies. We simply have to count them. As some of you might know, the primary sequence of antibodies is not disclosed for the most part, so we can't really tell if they're unique or not. So if you match these criteria, you're in what's called the ignorome or T-dark. Otherwise, you end up in T-bio, which means we have a decent understanding of the biology, but no chemical matter or drugs that modulate it. So why do we care about T-dark? It turns out that these were proteins like the leptin receptor or PCSK9 were truly dark proteins before 1995 or 2003 in the second case. And then a critical event happened, either receptor deorphanization or a strong protein disease association study. And then a few years later, typically 15 to 20, there is a drug on the market. So these are documented cases where you progressed from T-dark to T-clean in less than 2 decades. Of course, the road is arduous and it's not a linear. We can also tell you that there is a progression. So we started first this in December 2013. So that's the cutoff that you see there. About 90 to 100 proteins were in T-dark. And as you can tell, over the years, they progressed, keeping the same criteria to about 6,400. There's not as a dramatic improvement when it comes to T-clean. We had a slight revision in the criteria, so we had a dip there. But by the end of 2020, we were up to 659. The most recent count, which includes FDA-approved drugs for 2020, and we just submitted a short paper to Nature Drug Discovery, will add about 15 new proteins. So that number will be soon revised to be slightly higher. As I mentioned earlier, our resources are available through pharos.nih.gov. So if you go there, there are ways to look at this either by target or by disease or by ligand. Unfortunately, we still have not yet implemented the monarch disease ontology. We are observing the disease ontology implemented by Lynn Schrimmel at the University of Maryland at the moment. So I think that the first part of my talk was convincing enough to point out that we have a knowledge deficit. The number of dark proteins is steadily decreasing, whereas the number of approved drug targets is slowly increasing. Also studied very well and commented in the landmark publication of the IDG consortium is the aspect of the grantome. We analyzed all the R01 funded grants. The short version is if you're going to work on a dark protein or dark gene, the chances of getting funded are very, very small. So what do we do with all this information? We try to take it into account and focus on what's called protein disease associations. And for that, we use the resource maintained by Lars Jensen, diseases. And if you go to the disease database, there are 3 main channels: text mining, knowledge, and experiments. We are making, my group is making a direct contribution in the experimental channel through something that we call TIGA, which is Target Elimination by GWAS Analytics. And what that does is essentially a complex post-processing of the GWAS catalog and eliminating more accidental reports and rather focusing on those that are well cited and where the odds ratio is significant. So we try to clean up the GWAS catalog in a nice way. The other thing that we've done reasonably well is to implement a bidirectional encoder, BioBERT, and we text mine all of the PubMed abstracts plus the 2 million or so full text papers that are available in the subset of PubMed Central that is thus available. And this is done every week. So if you go to diseases on Monday morning, it's typically the latest version is updated with the latest information. When you look at that information, and in particular with respect to rare diseases, and Rare Disease Day is upon us, it's the last day of February, what we notice is this gap in translation. So if you just count the approved orphan drugs in Europe and in the US, you notice that it's about a 3 order of magnitude difference between those and the publications that come out every year. And this covers 3 decades of research from 1987 to 2018. So it's truly a situation that needs to be corrected, which is a segue to another type of research that we've done also with Melissa, Peter Robinson, and Chris Mangal, which was to re-evaluate the number of rare diseases. And this upset plot illustrates the overlap between NCI Thesaurus, Disease Ontology RD Slim, GARD, which is the NCATS database for rare diseases, Orphanet, and OMIM. And you can tell that complete agreement is observed on about 300. Presumably those are all the rare cancers. But other than that, there are resources that single-handedly look at rare diseases. And we, of course, looked at leaf nodes with respect to rare diseases. So that was all explained in the paper. So the other question that I asked was, well, what if we look at these proteins associated with rare diseases? Can we understand better the rare disease proteome? And as you probably noticed in the earlier circular histogram, TCLIN was at 3% and TKM was at 7%. This is roughly doubled on a smaller subset, and on the plus side, of course, T-dark is much less. This has to do with the fact that as we accumulate information about rare diseases, it allows us to do a better annotation of these proteins. So the majority of them end up in T-bio. And of course, if I were to do rare disease research on a daily basis, I would start by looking at the subset that's associated with T-CLIN, because those would be the low-hanging fruit. And in relationship with that, I was asked by the Mouse Phenotype Consortium, currently COMP2, but they're looking for the next 5 years of funding. The question is which genes to prioritize for the next 5 years. And the analysis that I started by was to ask the question, how many of the existing IMPC phenotypes are associated with rare diseases? And the answer is, and what's covered in this plot is about 2,500 genes. And this would be a really unique opportunity where you look at the mouse phenotypes and you ask the question, which of these phenotypes match with the phenotypes observed in the human disease? So it could potentially give us a wealth of models for disease research for each individual rare disease. So again, but other I promised in the title was machine learning. This is a complex plot, but just very briefly, we start by constructing a database. As mentioned, we look at disease associations, we look at expression data, pathways, protein-protein interactions, etc. And then we have scripts. Initially, these were done in R. We have migrated since to Python. And all that gets into a database, which then allows us to create models. So for example, if you start with type 2 diabetes, and if you have an existing set of genes in OMIM that are associated with type 2 diabetes, the script will generate that for you. You can also have an input that's manual input, or if you're looking at mouse phenotypes, this is also automatically extracted. So all these models are created automatically or by hand. And then the features are transformed according to the metapath concept, which allows us to do what's called graph embedding. So we generate knowledge graphs, which we then submit to XGBoost, and all that allows us to evaluate the results. And one way to evaluate the results is to look at variable importance. At variable importance in the case of diabetes, so I looked at this particular subset, which at the time had 51 genes. This was in September 2018. And I got a model. I checked all the proteins that were predicted by this model, at least the top 300 were all expressed in the pancreas. I even tried to look at beta-Langerhans cells, etc. I looked at hemoferritin and hemoxygenase. These are associated with a disease called hemochromatosis, which migrates to type 2 diabetes. So all in all, I thought I had a decent model. I sent this data and the predictions to Mark McCarthy, Mark was at the time leading the Type 2 Diabetes Consortium on behalf of the NIH Foundation and the Accelerating Medical Partnership Initiative. And as you probably can see from his email, they couldn't see any enrichment from my prediction to the genes that they were finding through GWAS or exome data. And then at the end, he was like, we're questioning the training set. You probably should not have used Omim. So the long story short, I got 54 genes from Mark and Anubha Mahajan, who was at the time his postdoc. I think most of this work has since been published in Nature Genetics, at least the genes that they used. And in the top 24 predicted genes from this model, there were already genes that were strongly confirmed by GWAS signal, as well as imputed GO terms, and the 2 out of the top 4 variables made sense from a type 2 diabetes perspective. So very different model, very different story. On the plus side, you can also combine this model with models that are associated with type 2 diabetes through the IMPC consortium. So these are 5 different models where we used, again, these were automatically generated by extracting all the positive genes associated with that particular phenotype, which is either related to glucose or insulin level, and then looking at the top predicted genes and then trying to evaluate how many of them are confirmed by GWAS. Again, this is translating from human phenotype to, from mouse phenotype to human phenotype. So initially the prediction is done on the mouse genes, of course, but then we look at the ortholog. And these are other genes that would be in support of the previous model that I showed. So I think it sort of illustrates the case that you can combine data across species and look at the different phenotypes. Another way to prioritize genes for the mouse consortium is to ask which are the truly dark genes. So this is what we call dark of the dark, if you think of chocolates. So currently in the data set that we track through Pharos and our system, TCRD, there's about 2,500 genes that are not well annotated in the literature. In fact, most of them are T-bio and T-dark. The majority of them are in T-dark. And of course, even in this subset, the vast majority of them do not have a predicted function. So these are also among those that we are considering for prioritization for the phase 3 of the mouse consortium. So the dark gene prioritization via IMPC, we cannot tease out causality. I think this is understood in the machine learning community, but the trick here is you need good data as input before you get to good models. And we assert that the IMPC negatives are true negatives because the way the mouse consortium works is they take their mice and they phenotype them, of course, across 540 different variables. And if no significant associations or changes are made compared to wild type, then they are not recorded. So if a gene that's been mutated lacks that phenotype association, that's more likely to be a true negative. So if you combine that with monogenic rare diseases, where you understand the phenotype, you can look at mouse phenotype and compare it directly with the human phenotype. And of course, you need to exercise care, but I think it would be a good rational strategy to prioritize dark targets. Having said that, I'm going to switch to cancer. This is an often asked question, how many cancer genes are there? And the answer is, if you look at the literature, as many as 2/3 of the human proteins have been associated with cancer. And if you step in with additional filters and you say, I want the publication score to be above a certain value, and we have ways to do that with z-scores, the short version is almost 25% of the human proteome is significantly associated with cancer. Does that mean they're good drug targets for cancer? No, I don't think so. A good subset to start with would be what's called the cosmic cancer gene census. And in the cosmic cancer gene census, you have a subset called the hallmarks of cancer. And I listed here the qualities that they have to follow, from proliferative signaling all the way to activating invasion metastasis. And the other set is what they call tier 1 genes, which includes the hallmarks plus a subset which I maintained separately for visualization purposes. And then a smaller set, which are genes that are more recently identified, and the evidence is not that strong, but it's strong enough to make it into their list. So all told, they have about 700 plus genes in their system. And I try to separate them. It would perhaps not surprise you if you work in oncology to notice that the kinase subset, which is this dark blue, dominates in the TCLIN and TKM. So most of those are kinases, but of course that changes when you switch to other categories. So we then use the same machine learning model with the knowledge graph embedding that I mentioned earlier and MetaPath, and ask the question, can we identify genes that are more similar to the hallmark of cancer, or tier 1 or tier 2? And then of course the question was which ones to use as negatives. So what we used as negatives, at least in the first model, which I think is more strict, was housekeeping genes. And this was a notion that was proposed almost 2 decades ago, but we also looked at additional data from 2 separate sources and confirmed that these are housekeeping genes, and also eliminated additional criteria such as they shouldn't have been associated with cancer in the primary literature, and we looked at Lars Jens's system. And this is just very briefly how the models fare. And we're going to focus on this one because it has the best, when you look at cross-validation, it has the best confirmation. So this is the confusion matrix. And of course, what you want is the positives to be as large as possible, closer to the original, and the negatives should be as close to the original as possible. These models sort of trail off, so I'm not paying a lot of attention to those at the moment. The other thing that I looked at is the feature selection, and the top features that are selected is from the cancer cell line encyclopedia and the protein-protein interaction notes from String. So how does the cancer cell line encyclopedia look like? This is a T-stochastic nonlinear embedding plot where you take expression data from almost 1,400 cell lines that are relevant to cancer and about 16,000 genes. And what you can tell is that all the blood cancers are nicely clustered on one way. Male lung cancer are here in the bottom. Female breast cancers are here. Colon, colorectal are here. So it does give you some perspective that some of these cell lines are very well differentiated when it comes to things they have in common with respect to gene expression. Unfortunately, not everything is as well delineated, and I tried to put in things that sort of overlap within the middle. And of course, that's always a problem with research, that you still dig into the data as much as possible. We're looking at the same plot, but this is now clustered by protein function. The most interesting part here is the olfactory GPCRs, which are totally underexpressed, and they only cluster in a very small subset. And if you compare that with the non-olfactory GPCR, it's a very different story. But this is the most interesting cluster. The other one that might be is the epigenetic regulators. I also had hoped that nuclear receptors and transcription factors would give me some clue as to modulating cancers, but they do not. So to be continued. But then how do we take this CCLE data and analyze it? So when I look at the top 100 predicted genes with the hallmark model, I can get 16 of the tier 1 reproduced by the model, which is not a bad thing. And overall, 76 out of the 100 through text mining already have association with cancer. So these would probably be good candidates for cancer research. And of course, after I eliminate in the top 30, I eliminate 11 of them, which are already in the tier 1. these are the genes, and 4 of them are kinases, and 1 of them is an olfactory GPCR. And I actually double checked on that one, and it looks intriguing to say the least. So to conclude this part of the talk, I think you can use machine learning to prioritize genes, and it says here cancer, but you can imagine if you have enough data, it could be pretty much any disease from diabetes to Alzheimer's. But of course, what we really need is support from the scientific community, because in order for us to validate models, that takes a lot of effort. So the way I expressed it is that we can generate computer models a lot faster than the community can validate them. And it's really work in progress, and hopefully we'll get some people to act on this and validate. I also wanted to put in a thought about beyond the simple idea of targeting proteins and nucleic acids. I think there are additional metals, lipids, stem cells, sugars, perhaps other entities that should be considered non-traditional drug targets. And we sort of think of drug target in a boxed way. We target a gene, we target a protein, and those are typically what make the cover of Nature and Science. In reality, drug discovery is probably a lot more complex. Oligonucleotides are currently making a strong comeback, monoclonal antibodies, etc., plus stem cell therapy. So a lot more work to be done. I don't know how I'm doing with time. I have another set of slides, so I could continue or I could stop here. It's up to you.
It's about 12:05. People typically, leave maybe 10 or 15 minutes for questions. So if you have something you could cover in the next maybe 10 to 15 minutes, that'd be fine.
Okay, so I'm going to quickly pass through these things and just show you a little bit about COVID research. I'm going to share the slides, so if you have questions, you can go through them and ask. So the story with COVID, which probably interests everyone, this is a drug repositioning story. I found it very confusing, and this slide belongs to Avi Mayan and his group. He published this. He collated information from a lot of publications, and the only thing they seem to agree upon is that remdesivir seems to work in 7 out of the 12 papers they looked at, but it doesn't work in all of them. So the real difficulty is, of course, experimental confirmation. And it goes back to the old saying that if you have a clock, you can tell time. If you have 2, you cannot tell time with precision. And that led to this comment that we landed in Nature Biotechnology, where we tried to point out to the fact that publications by Twitter should not be considered scientific publications. Hopefully all of you know what I mean. Hydroxychloroquine is not a good drug for COVID. And a lot of papers were published strictly on computational predictions of bioactivity, which I found clouds the field. So we made extra effort to confirm that independently, which I will show later. And part of our argument was that as a community, we have to take action against the chloroquine story, lest it becomes the same story as vaccines cause autism, which the paper was debunked, it was retracted. Andrew Wakefield lost his medical license, but it was too late. It created a movement. So what we did back in March, at the time we thought chloroquine was active, but so we started with chloroquine as a template. It has 2 isomers, R and S, so we used both of them. This is about 3D modeling, and we identified using molecular interaction a set of drugs that we submitted for experimental testing. And the compounds that we found to be quite potent was amodiaquine, which is an antimalarial drug. In our hands, it was a sub-nanomolar potency when it comes to the cytopathic effect, and zuclopentixol, which is an antipsychotic. We then independently submitted this to experimental confirmation to the University of Tennessee Health Sciences Center. So the group of Colleen Johnson did work independently. We bought a separate batch of each chemical and we sent it to her. So we had two groups that were confirming it. In this case, with amodiaquine, the experimental error was about 2 orders of magnitude, but it was still active. And this has to do with different experimental conditions. More importantly, what we found is that the active metabolite of amodiaquine is also potent. And the neat thing about this one is that it has a high concentration in the serum, the Cmax, and its half-life is 500 hours, which is 3 weeks. So our experimental results, which is where I show that chloroquine doesn't work. So this is chloroquine. This is an assay called viral titer reduction. So essentially you expose the cells to the drug and then you infect them with increasingly higher concentration of the virus and you look at the protection. So it's the equivalent of a dose response, but a fixed dose of the drug and an increasing dose of the virus. And then you're looking at how many orders of magnitude it protects against the virus. And the short version is that chloroquine doesn't protect, but remdesivir and this active metabolite of amodiaquine have a similar protective effect. We also found a protective effect from nebivolol, which is a beta blocker, and zuclopentixol. And it's not our work. I found this in bioRxiv. This is experimental work in hamsters. Very briefly, amodiaquine protects against infection. Amodiaquine protects against transmission. And if your animal is infected and another animal is given the drug and exposed to the infected animal, the animal that got the drug does not get the infection. So this is pretty much confirming what we found, but with a much bigger scale because they had the money to do the experiments. So we think that amodiaquine could be serve as backup for remdesivir, considering that remdesivir is $2,500 per cure, whereas amodiaquine is 5 rupees for 10 tablets, and you only need 3 tablets for 3 weeks, and you're good to go. This would be a much better option for the countries that cannot afford or cannot access remdesivir. A shout out to a machine learning effort that just got accepted on Monday morning to Nature Machine Intelligence. So we took data from NCATs and we built a suite of machine learning models, which once you input the chemical structure, and there's 3 ways to do that. It standardizes it and then it predicts viral activity against COVID. And the short version is that we can predict these bioactivities. This is the output from Redial 2020. And in this case, so NCATs had not tested desethylamodiaquine, but it was predicted to be active. So in some way, at least, we confirm our own findings. And we also offer on the website ways to clearly interpret what the models mean and how to be used. And this paper was in peer review for more than 180 days, so it was gruesome. So I guess drug repositioning is an iterative exercise, and you need multiple experimental confirmations from independent groups in order to progress to clinical trials. So I'll stop here and look forward to questions.
Great, thank you. People either can type questions in the box in the chat, or they could unmute themselves. I will I actually have a question for you. And I also want to thank you for reminding me that you took the 10 by 10 course, which many students in our list here will know locally as BMI 510. And I'm glad that I contributed that to your knowledge. One of the actually, I was thinking as you were talking about the drug for COVID that is really cheap. It's great to identify drugs like that, and of course, there's a lot more to identifying it in a model. In fact, that reminds me as well that many of the preprint, because of this proliferation of model papers, a lot of the preprint servers have stopped taking model papers. But my question really is, how often do we, truly discover better or even comparable new drugs by these methods? I certainly, we can do a lot of computation now. We can identify genes, we can identify proteins associated with diseases that are potential candidates for drugs. But knowing what I do about clinical research, there's a lot more to it than just identifying the drug and carrying it out into phase 1, phase 2, randomized control trials, and so forth. Are there how often does that happen? Are there good examples you could point to show how this process, and the computational sides leads to medical advances?
So right now, this is a controversial topic. Al Edwards, whom I mentioned earlier, Structural Genomics Consortium, just published a commentary in September last year, pointing out that we don't really get a lot out of drug repurposing papers. All we get is a claim, but it's not followed by the clinic. So I've been spending time, the little that I have, I was just looking at PubMed, the subset that is the clinical subset, and then that's about 2,500 papers. I looked at those that have clinical trials, and it says that there's 140. And then if I manually select those that might be interesting, the answer is 29. So I have to look at those 29 papers in more detail to figure out how many were successful. But I'd say I could probably count them on my hands. So I wouldn't say there's more than 10 that were truly repurposed. And that doesn't, I'm not talking about sildenafil, which everyone knows about, and drugs like thalidomide that made it on the market. So those, of course, are well-known examples, but those did not come from academics or high-throughput screening or virtual screening for that matter. So I'm specifically looking for those that were contributed. So I'd say not a very large number for all the noise that the field currently makes.
Mm-hmm. There are some questions in the chat box. Actually, if you, it's usually easier to read if you go off of screen sharing. When you go on screen sharing,
okay, so stop sharing. Okay, I just did that.
And in the chat box, there's some questions.
Okay, so what is, Robert Dodier asks, what is the metric for assessing variable importance? So I'm strictly looking at the gain function and the cumulative gain function across 100 cross-validated models. So I'm looking at the contribution across multiple models where you do cross-validation. I hope that answers the question. And the non-human targets, that's strictly, I mean, we get infections, whether it's virus, parasite, bacteria, so anything that's not a target is a non-human target. So we need to drug those too. So that's the answer to that question. Are there any other questions?
Hi, Tudor. This is Guanming. Thanks for the great talk.
Hi, Guanming.
Yeah, so I have a question. So in your machine learning approach, you basically build a big knowledge graph, right? So from
Yeah.
your experience, is it like always true if I have a bigger, so like basically I can collect all this knowledge into single graph. So will knowledge graph the bigger, the better for the actual machine learning output or performance? Is that your experience?
Good question. Bigger doesn't mean better. Obviously, you add noise. So what we've started doing repeatedly is to perturb the model by swapping 10% of the positive labels with negative labels and vice versa, and looking at stability of the model. And the other thing we do is we routinely eliminate the so-called large data sets like the CCLE and the links, the cellular signature perturbation networks. So we build models with and without links and CCLE. So we essentially generate 4 models, one with everything, one without links, one without CCLE, and one without links and CCLE. And then by doing this multiple perturbations, we then ask the question, how many of the predictions are stable in 80% of the models? And then we look at the comparison. So that's what we've ended up doing lately. Does that answer your question?
Oh yeah, thank you.
Yeah.
I guess maybe one other question I can ask. He's actually not here, but another member of our faculty and I have been looking at diagnosing rare diseases through processing electronic health record data. Are there any, I'm trying to think of how you might use clinical data, patient data. It sounds like most of your work is focused on literature data. Are there ways that you could, I'm just trying to think, co-occurrence of diseases, people who, patients who have had their genomes sequenced or things like that. Are there any opportunities for clinical data from patients to complement?
Yes, we do that as well. So we routinely mine Cerner Health Facts, and we routinely mine the Truven Market Scan, which used to be Truven, it's now IBM. So we look at those, and of course there's limitations with each of these data sets. With Cerner, we still have a problem that it's not following the OMAP data model, so we still have a problem with that. But we essentially look frequently at comorbidities. We take Loin codes and look at certain biomarkers like blood, serum levels, whatever available, and ICD-10 and stuff like that. Don't have anything that's primarily driven by myself published, but for example, in 2012, I was in Denmark at the time. So we have a paper in Nature Communications where we looked at the temporal disease trajectories across the population of Denmark. So 15 years worth of disease data and looking at temporal relationships. So that was it's a well-cited paper.
It's another question in the chat. Looks like it may have gotten cut off.
Yeah,
I assume as a treatment.
So amodiaquine is being studied as a treatment by folks at Mount Sinai and Harvard, but it has not yet made the cut to the NIH adaptive trial system. I was looking at most recent announcements there, and they are not yet looking at that as an alternative. And I guess part of the problem is that it was approved in the US, but it's no longer approved in the US. So it's what you call an off-market drug. And so it's the question is a bit more complex. So the best way to study this would be to go to countries like India, where it's easy to find it. I should mention that amodiaquine is on the WHO list of essential medicines, so it is widely available, it's widely understood, it's used against malaria, so it would be a simple effort to repurpose it.
Well, okay. We doesn't look like we have any more questions. So thank you for giving your talk. It was very interesting to hear what you're up to, and maybe someday we'll get you to Portland in person when we
That
can
would be
start
nice.
traveling.
I would appreciate that. Yeah. Thank you for inviting me.
Yes, thanks for coming. Okay.
Okay. Yeah.
See you all
Take care.
later.
Okay. Bye.
Bye.